All right, today we're jumping headfirst into a real-life, scientific detective story. We're going deep into our own genetic code to uncover the roots of conditions like autism and intellectual disability, and we're following a team of researchers on an incredible quest for answers.
So let's kick things off with a pretty big question, right? What if the key is to understanding something as complex as how our brains develop were hidden right there, inside our own DNA? Well, this isn't science fiction anymore. It's a mystery that geneticists are working to solve as we speak.
And trust me, this is a deep mystery. For a long time now, we've known that neurodevelopmental disorders have a really strong genetic link. We're talking heritability estimates that can be as high as 80 or even 90%. But here's the thing, for most families, the specific genetic reason why has been an agonizing unknown.
So, just to make sure we're all on the same page here, the term we're going to be using is neurodevelopmental disorders, or NDDs for short. It's really an umbrella term for a group of conditions, like Autism Spectrum Disorder and intellectual disability, that all have one thing in common. They affect the development of the brain and nervous system.
And this is where our story really begins, with 100 families from a specific region in Italy called Valle d'Aosta. For them, this isn't just some abstract scientific question, you know? It's a deeply personal search for answers, and they became partners in a really groundbreaking study.
So, what's the game changer here? What new tool do our genomic detectives have that lets them crack these kinds of cold cases? Well, the answer lies in a totally revolutionary new way of reading our genetic code.
For years, the go-to tool was something called Whole Exome Sequencing, or WES. Think of it like a basic roadmap of a country. It only looks at about 1% of your DNA. Just the major highways, the parts that code for proteins. It's useful for sure, but it misses a ton, leaving about 70% of these cases completely unsolved. But now we have Whole Genome Sequencing, or WGS. This isn't just a roadmap. It's like upgrading to a super high-res satellite map of the entire planet. It scans over 99% of your DNA, every highway, backroad, and footpath. You see everything, and that incredible detail can boost the chance of getting a diagnosis by at least 8 to 10%.
Okay, so armed with this incredible new tool, the researchers got to work, setting out to map the genomes of all 298 people in the study, the kids and their parents. But this was no simple task. They were about to be hit with an absolute tsunami of data.
So how much data are we really talking about here? Well, get ready for this. In their very first pass, they found more than 26 million unique genetic variants across the group. I mean, that number is just staggering. The one critical clue they were searching for was buried somewhere in all of that.
And this is where you really start to understand the whole needle in a haystack problem. Just look at this chart. Over half of those millions of variants, 54.4% to be exact, were found in what are called intergenic regions. That means they're in the massive, sprawling spaces between our genes, the exact territory that the old roadmap method would have completely missed.
So how on earth do you find the one critical clue in a haystack of 26 million? Well, you filter. You act like a detective. The scientists ran all that data through a whole series of filters. They'd ask, is this variant super rare? Is it in a gene that we know is important for brain development? Does the way it was inherited in the family make sense? With every single step, they narrowed it down, shifting millions of potential clues down to just a handful of prime suspects.
And after all that bigging, all that filtering, all that detective work, they had a breakthrough, a huge one. For so many of these families, after years and years of uncertainty, they finally had some answers.
So what was the final number? A diagnostic yield of 26.4%. Now, let that sink in for a second. That means for more than 1 out of every 4 families in this study, after all that searching, they found it. A likely genetic cause for their child's condition. This is so much more than a number. For these families, it's an answer that can finally bring an end to a long and often painful diagnostic journey. But here's where it gets really interesting. When you look closer at that number, you see the success rate wasn't the same across the board. Far from it. As you can see here, for individuals with just intellectual disability, the diagnostic rate was an incredible 44.4%. For those with both autism and ID, it was 25.6%. And for those with autism only, it was 15%.
And that difference? That's a huge finding. It's basically telling us that whole genome sequencing is an absolute powerhouse for finding answers in cases of isolated intellectual disability. The thinking is that these cases might be more often caused by a single, really impactful genetic variant, and that's exactly the kind of smoking gun that a full satellite map of the genome is designed to find.
You know, to really get a feel for how this technology can solve puzzles that used to be impossible, let's zoom in on one specific case, family NED029. It's a perfect example of just how complex these mysteries can be.
See, in this family, it wasn't one single genetic smoking gun. No way. What WGS found was that the child, who was diagnosed with both autism and ID, had inherited this incredibly complex cocktail of genetic changes from both parents. From their mom, a large duplicated piece of a chromosome, and from their dad, 3 other big changes, a couple of deletions and another duplication. It was the combination of all these different pieces that likely led to the diagnosis. There's just no other technology that could have seen all these different types of variants and pieced that intricate puzzle together.
But what about the other families? You know, the other roughly 3/4 of cases. For them, the files are still open, their genetic cause still a mystery. But the story doesn't end there. In fact, this is where the investigation takes a really exciting new turn.
The single biggest hurdle in this kind of work is something called a variant of uncertain significance, or a VUS. Basically, it's a genetic change, but scientists just don't know what it does yet. Is it harmless, or is it the clue they've been looking for all along? For our genomic detectives, these are the unsolved clues, the smudged fingerprints they can't quite identify, yet.
But now, our detectives have a brand new partner on the case: artificial intelligence. Seriously, the researchers used an AI model to go back and re-examine all those uncertain clues. This AI has basically been trained to recognize the super subtle, complex signatures of variants that we know cause disease. And when they let this AI loose on their cold cases, bam! It flagged a specific VUS in a gene called KALRN as a super strong suspect. This is a huge deal. An AI-driven approach like this could be the key to turning a whole lot of these question marks into definitive answers.
And that just leaves us with this incredible forward-looking thought, right? You've got the complete satellite map of the human genome. You've got a powerful AI partner to help you read it. The toolkit our genomic detectives have now is more powerful than it has ever been. So the only real question left is, what mysteries are they going to solve next?